Welcome, everyone. I'm going to speak in English because, unfortunately, I have no Portuguese. I hope that you can understand me. And can you hear me in the back? Okay. Let's raise this a little. Is that better? Okay, great. So I'm going to talk about queering herpetology on human perspectives and the study of diverse animals. And first I want to thank Felipe Toledo for inviting me, and particularly for inviting me to give this talk. I'm very happy to be here in the Bertha Lutz Auditorium in Campinas at a conference whose theme is inclusive herpetology in 2019, which is the 50th anniversary of the Stonewall uprising. Giving a talk titled Queering Herpetology. That's kind of amazing. And for some of you, what I just said makes sense and may resonate in ways that perhaps I can't even imagine. And for some of you, queering herpetology is not something you have any idea what it could possibly mean, but you're curious. Maybe some of you just want some embryo eye candy. I thank all of you for being here. So as Guilherme said, I'm an integrative and comparative biologist, and I'm interested in phenotypic plasticity and development and behavior and critical transition points in animal lives. And I work mostly on frog eggs, and I'm known for my discovery of predator-induced hatching and integrative work on environmentally cued hatching. I also work in the new field of biotromology, so trying to understand how embryos make life and death hatching decisions using incidental cues from predators against the shaky vibrational background of life in a blob of jelly on a leaf in the rainforest. And this has been an 18-year collaboration with vibrations engineer Greg McDaniel. Since 2011, half of my teaching has been in gender and sexuality studies, in collaboration with humanists and social scientists. And I bring things that biologists know about diversity and plasticity into interdisciplinary conversation with work from other fields, and I bring insights from gender studies back to biologists. And more recently, I've also tried to meld these two sides of my life and of my career for general audiences. So here's an outline for my talk. I'll first define some terms, then I will review some scientific and social history. I'll introduce the concept of heteronormativity and consider what its effect is on what we know about animal behavior. I'll consider scientists as mammals, studying much more diverse herps, and then I'll look at what we know from social science about the value of human diversity for our understanding of the world. I'll look at also what we know from social science about queer folk working in science, technology, engineering, and math, and I'll share some personal history from my life, and end with some thoughts on the value of weird observations. Okay, so first definitions. Queer is an unstable and multifaceted concept that different people use in different ways, and in the humanities, that's part of its value. It appeared in English early in the 16th century as an adjective meaning odd, strange, unusual, perhaps deviant, suspicious, a little questionable, and also slightly unwell, in opposition to ordinary and normal. It was also used as a verb for to spoil or disrupt or undermine. Later, it came to be used for people, particularly meaning homosexual, but also more generally outside the norms and conventions of expected sexual behavior or gender expression. In this sense, it was used, and sometimes still is, as a slur and some people find it offensive. As a biologist interested in variation, I like these old meanings of unexpected, perplexing, weird. Queer can be something worth understanding better. Starting in the 1980s, queer was reclaimed as a word to celebrate human diversity, to positively claim non-normative or marginal desires, and to make space for variant sexual perspectives. As what had started as the gay liberation movement, aiming for social transformation, increasingly focused on obtaining rights such as marriage for gays and lesbians who might fit into slightly expanded social norms. So queer still has this non-normative meaning, but it's also now used as an inclusive or umbrella term for gender and sexual minorities. So lesbian, gay, bisexual, trans, questioning, intersex, asexual, pansexual, non-binary, gender non-conforming. There are so many different identity variants these days. So queer is useful as an inclusive umbrella term for this diverse and expanding community of people, and it doesn't force us to draw lines between potentially fluid or shifting identities. As a verb, queer can mean to apply non-normative perspectives, particularly the critical methods of queer theory in the humanities, to concepts, academic fields like herpetology, etc. It can also mean to include and welcome and integrate gender and sexual minorities into a field like herpetology, and the changes that may result from our presence or increased visibility. So today I will use queer broadly to include both gender and sexual minorities. Sometimes we separate queer sexuality and use a trans umbrella concept for genders. So it's also relevant that normal has a variety of meanings, from ordinary and conventional to healthy and natural. And people see themselves and others in relation to these social norms. And this idea is applied to individuals and to the traits of individuals. But it's also fundamentally contextual. So, for instance, because I grew up speaking English, which uses egocentric spatial references, here I think the eggs are in front of me. That's normal. But if I had grown up speaking some other language that uses geocentric spatial references, here it would be normal for me to think the eggs are northeast of me. And I would not have had to look that up on Google Maps. So this contextual and often value-laden sense of normal is a subject of critique and analysis in the humanities field known as queer theory. Now, as biologists, we often think of normal in terms of distributions, and we're interested in understanding all the variation. There's, of course, not one best way to do this. There's many ways, but it is clear that different perspectives, different foci, different sampling strategies will give us different information. And too narrow a perspective or biased perspective could mislead. So we think a lot about this when we're designing experiments, when we're evaluating experiments, but perhaps less when we're choosing research questions. But of course we know that our understanding of life has accumulated through the questions that people asked and put the work into answering throughout the history of biology. This informs and frames the questions that we ask today. And our thinking is shaped by the societies and cultures that we live in. And so this affects what people can even imagine to be possible. For instance, in the Victorian era, when Darwin proposed sexual selection, as an evolutionary mechanism to explain the origins of male traits disfavored by natural selection, the idea that males compete for mates and the winners pass on their competitive traits, that made sense to people. The idea that females actively choose mates and therefore their aesthetic preferences drive the evolution of male displays, that was really hard to imagine in Darwin's day. And it wasn't until the 1970s when Trivers proposed parental investment in a much different social context that biologists began making substantial effort to test for female choice, which of course has now been amply demonstrated in many, many taxa. And then the paradigm of the eager, undiscriminating male and the coy, choosy female was overgeneralized on the basis of too little evidence. And we're still affected by that overgeneralization today. Even though, in the same era, women field biologists, including Sarah Hrdy and Patty Gawadi, were noticing that their study species didn't fit this paradigm. So females actively sought sex, sometimes from multiple males, sometimes even when they couldn't conceive. So things were more complicated. Things are more complicated. And bringing feminist perspectives into evolutionary biology transformed the field, and it changed how we understand ourselves and how we understand other species. In the 1969 Stonewall uprising, remembered around the world this year, queer people in New York City fought back against a routine police raid. At the time, same-sex relations were illegal in the US, gender conformity was actively policed, and people could lose their jobs for being gay, which, of course, in many states, they still can. In my country, Canada, Pierre Trudeau's government had just decriminalized same-sex relations, along with contraception and abortion, declaring there's no place for the state in the bedrooms of the nation. And this Stonewall uprising, of course, wasn't the start of the fight against gay oppression, but it was a catalyst and a turning point for public organizing. And in the half-century since, social recognition and treatment of diversity in human sexuality and gender has changed considerably. So among countries, the current laws vary from constitutional protection to the death penalty, with a whole range of things in between. And if you want to know more or look up the laws in a particular country that you might be thinking of going to for field work, go to ilga.org. Okay, so the word heterosexism means inequalities that privilege heterosexuals and discriminate against sexual minorities. And so, for instance, the Tanzanian law that mandates life in prison for people like me is an example of structural heterosexism, right? It's built into the laws of the country. But heterosexism can also take more subtle forms, like unconscious biases of well-meaning people. And here, the concept of heteronormativity, I think, is really useful. So this is the routine assumption of heterosexuality and associated behaviors, family structures, life history attributes, etc. It tends to make certain kinds of variation invisible, while visible difference is odd or queer and needs to be explained. Meanwhile, normative traits are naturalized, unquestioned, and unexamined, right? So we don't understand them for other reasons. So this, of course, affects how we see or don't see queer families. So for instance, Ondine is not obviously recognizable as my partner in many contexts. but it also affects many other families. For instance, Brad is easily recognizable as my brother in most contexts, but Peter is not. I have to tell people. Heteronormativity can also affect our observations of other species, what we notice, what we record, how we interpret what we see, what we publish. So we tend to interpret courting or mating animals as male and female, but they aren't always. So in these Thamnophis, cold males emerging from hibernacula temporarily release female pheromones, attracting courtship from warmer males, which helps the cold ones to warm up and also protects them from predatory birds. And these kinds of phenomena may often go unrecognized or unnoticed. Same-sex sexual behavior is now documented in many hundreds of species, typically by individuals who also mate heterosexually. It's estimated to occur in about 15 to 30% of mammals and birds, but we really don't have good estimates yet for the incidence in amphibians and reptiles. There can, of course, be many non-adaptive reasons for any behavior, so same-sex mating could occur simply as a side effect, an incidental side effect of other adaptive traits. But of course, once a trait is expressed, it's available for selection, and there's now substantial evidence for non-reproductive functions of sexual behavior, including in same-sex contexts. And really, this should not surprise us, because it's well established that traits that evolved in one context can later gain new functions. And the ancestral function may be retained or sometimes lost. And the discovery of sexual behavior in same-sex contexts where its function cannot be fertilization creates opportunities for research that can improve our understanding of sexual behavior more generally. So these lizards are both female. They're members of the all-female Nematophorus uniparens. And David Cruz and collaborators noticed this behavior, the pseudocopulatory behavior, in the lab, and they documented that it increases fecundity. And then they went on to do an elegant series of experiments comparing this species with their sexually reproducing relatives to work out the neuroendocrine mechanisms underlying this behavior and reproduction and how they've changed evolutionarily. And this has broad implications for our understanding of vertebrate sexuality beyond this unisexual species. So same-sex sexual behavior is, in fact, just a small part of the queer diversity of life. And it might not be where herpetology has the most to contribute. So one of my earliest herpetological memories is catching chameleons in the backyard as a kid growing up in Kenya, because all the other lizards were way too fast for a 6-year-old. But chameleons are also amazing because they change color, and even to a 6-year-old, this is obvious, and it was clearly important in their biology. So, of course, people have studied this, and there's great papers on visual communication in chameleons. Chameleons might also use substrate vibration to communicate, but we're not sure, because in the 20 years since this vibration recording and the initial behavioral observations were published, it seems no one has done the playback experiment to actually test this. Why not? Well, I'm standing here talking to you and showing you slides because sound and vision are how humans communicate. And if we look at research across sensory modalities, there's a lot more work on acoustic and visual communication than on olfactory and vibrational communication. And note the log scale. this difference in effort clearly does not represent their relative importance in the lives of all the other animals. It is, however, congruent with human sensory biases. So does human biology bias our study of and our understanding of sexual and reproductive biology? And can herpetology help? Well, so we're mammals, and as such we're gonochorists, not hermaphrodites. We have XX, XY sex determination at conception following internal fertilization. We're viviparous and placental, and we have maternal care with lactation. And most mammals share these traits, with a few less well-studied exceptions, even though they're super diverse in all sorts of other ways. Okay, what about birds? Again, while some traits are diverse, many aspects of sex and reproductive biology are shared ancestral traits, which doesn't give us material for comparative analyses. Herp reproductive diversity, as we all know, is a lot higher, but also less studied. Okay, for one example, consider parental care. There are more papers about parental care in mouse, that's not like all the mice, that's mouse, than there are in frogs. Okay, and there's so many more on birds where it's obligate and it evolved once. If we look at frogs, we think that care occurs in about 10 to 20% of species. It's super diverse, evolved many, many times, so we have fantastic opportunities for comparative work. Male, female, and biparental care are all widespread, especially in terrestrial breeders. And there's no sex bias in care duration or the level of protection that parents offer their young. So looking at this, the idea that anisogamy somehow necessarily leads to female care, it seems empirically unconvincing. For a lot of frogs, though, of course, we know very little about their reproductive biology, right? So our understanding could change. Let me give you an example. So Jesse D'Elia studies glass frogs, and a few years ago when he started his PhD with me, we thought about 20% of glass frogs had parental care, and we thought that it was always by males. Then, with Laura Bravo Valencia, very late at night, they noticed females sitting on eggs, just for a few hours. So might this matter? Well, through lab and field experiments, Jesse and Laura found that this brief period of maternal care and the hydration that mothers provided during this time was absolutely essential for both protection from predators and avoiding egg dehydration. And Jesse and Laura's field work, which they did a lot of, completely changed our understanding of parental care in this whole family. Right? So now it seems that all glass frogs have and all glass frogs need care. And male care has evolved 3 times from ancestors with female care. And every time that males took over caring, it became longer and more elaborate. And male care is the longest in species where males mate multiply, and among individuals, care duration increases with individual mating success. So minimalist mothering and elaborate paternal care again run counter to expectations from traditional conventional sexual theory. And the variation in glass frog parenting offers great opportunities for us to ask new kinds of questions. If we look across topics, reproduction, life history, sex determination, sexual development, there's been less effort into understanding these things in amphibians and reptiles compared to birds and mammals. But of course, it's more diverse in amphibians and reptiles. And looking at this, what I wonder is how much reproductive, sexual, and life history diversity in herps is still undiscovered. Probably a lot. We just saw how many species are undiscovered. What they do is certainly undiscovered. But even species that we know, we don't necessarily know what they do. Another example, my first PhD student, Justin Touchon, works on Dendropsophis imbricatus, in one of over 50 frog lineages that evolved terrestrial eggs. And one day he decided to collect eggs at this pond, where he hadn't worked before, but he knew that there was a good chorus. Surprisingly, he found no clutches. So this was weird, and so we went back at night, and we saw this. These terrestrial breeding frogs were laying their eggs in the water. And this simple observation opened a lot of questions and research opportunities. So in experiments, Justin found that ovipositing females and pairs assess local conditions and make plastic behavioral choices between putting their eggs in the water and putting their eggs on land. And selection pressures vary among ponds, so eggs do better on land in some places at some times and better in the water at other places and other times. And in comparative work, Justin found 5 other species of Dendropsophis in orange that show similar plasticity in aquatic and terrestrial ovipositions. And phylogenetic support that this plasticity may play a role in transitions from fixed aquatic breeding to fixed terrestrial breeding. And he also found variation in Dendropsophis imbricatus populations, including some places where they only lay eggs on land, and also individual variation in this population that he's now using as a basis for a selection experiment that may eventually tell us a lot about how terrestrial reproduction evolves on a genetic basis. So all of this from just this one observation of, like, there should be eggs here. Where are they? Okay, even in relatively well-studied areas, like sex determination in reptiles, there's still lots to learn. So we know there have been many origins of different kinds of genetic sex determination in reptiles, and many origins of environmental sex determination, including some very recent evolutionary transitions, which of course are great to study. For instance, in this skink, in high altitude populations, sex determination is genetic. At low altitudes, it's temperature dependent. And this divergence is adaptive given the climates that they live in, as Penn et al. Showed very beautifully a few years ago. So these kinds of population comparisons can tell us a whole lot about how and why different sexual and reproductive traits evolve. More recently, Haleli et al. Found widespread evidence of sex reversal, so from ZZ males, genetically, were phenotypic females in the wild in Australia. And sex reversal may often play a role in transitions between different sex determination systems. And these sex-reversed animals were viable, and when they were mated to ZZ males, they produced viable, fertile offspring. And so in one generation in the lab, they completely switched from a genetic to a temperature-based sex determination system. And in the wild, there are still lots of ZW females, but of course the climate is warming, so this may be indicative of changes to come. And of course, understanding why sexual reproduction is so common, given the immediate advantages of asexuality, is a problem in evolutionary biology. And there are over 40 lineages of obligate parthenogenesis in squamates, most but not all from hybridization. And the all-female Ambystoma salamanders have persisted for 5 million years, so way longer than us, reproducing asexually by gynogenesis, using heterospecific sperm to activate the development of their eggs, and occasionally incorporating a bit of sperm DNA, which we call kleptogenesis. And since 1997, reports of sporadic parthenogenesis in species that normally reproduce sexually have been accumulating, most with loss of heterozygosity, but some clonally, most in captivity, but some in the wild. And so the boundaries between sexual and asexual reproduction that once seemed very rigid are seeming increasingly permeable. Okay, so I've reminded you of something that I'm sure you all already know, that herps are far more diverse than mammals in terms of their reproductive and sexual traits. And we know this despite the fact that we've invested less effort into studying this. And I suggest that both our mammalian biases and the ways that human social history has shaped the progress of science have slowed our discovery of reproductive, sexual, and life history diversity in these and other animals, thus limiting our understanding of life. And I think that more active inclusion of human diversity in science might help speed up our progress. And we can benefit from the ways in which humans are actually more diverse than most other species of mammals. So let's consider that. So comparative primatology shows a 45 million year history of non-reproductive social functions of sexual behavior in anthropoid primates, and a 30 million year history of bisexual capacity and behavior in catarrhines. And all the great apes, not just bonobos, show both same-sex and heterosexual behavior. So we inherited an ancient capacity to use sexual behavior in many different ways for many different reasons or purposes and with different kinds of partners. Then our ancestors evolved cooperative breeding, involving shared childcare of very, very needy young. And this radically changed both environmental effects on development and natural selection on human variation. So as well as diversifying family structure and the organization of childcare, cooperative breeding probably contributed to increased genetic and phenotypic diversity in human gender and sexuality via socially contingent costs and benefits of individual reproduction and greater potential for kin selection. And if you're interested in this, please look up Paul Vassey's work. So what this means is that while other apes and catarrhines more generally have an expansive capacity for diverse sexual behavior, humans show more variation in gender and sexuality traits, both among individuals and also more variation among cultures. And this is part of the richness of human diversity that we have to draw on, right? And I say to draw on because social science has shown many benefits of human diversity. So just a lot of this work has been done on business and other fields, but just in science, citation rates are higher for mixed sex teams than all male teams. Also for ethnically diverse teams versus single ethnicity teams. Also for international collaborations versus collaborations within the US or collaborations within the UK. Plus, scientists who have lived in different countries have higher impact factors. And this is including people who moved with their families as children, not just people who had prestigious international fellowships. And these benefits of diversity among people come from variation among different individuals, like gender or race, and I would argue sexuality, but also within individuals, such as international experience or bicultural heritage or multiple social identities. And there's a lot of social science on this. And diversity improves creativity, It improves complex thinking, problem solving, a whole bunch of useful things. And really, really importantly, this benefits everyone. So interacting with people who are not like yourself has performance benefits both for majority group members and for minorities. But these positive synergies can be blocked by both overt discrimination and by more subtle unconscious biases, right? So we need to work on those. And here, in kind of translated into graphical form, the value of token diversity without active inclusion, or the presence of people whose differences are invisible or suppressed to fit in with group norms, is limited compared to open interaction among people who are different and free to be themselves. Okay, so we know that women and certain ethnic and racial minorities are underrepresented in STEM fields, science, technology, engineering, and math. And documented inequalities contribute to this, and in many places there are active efforts to improve equity and inclusion. And this is because science needs to tap all the talent out there, all the human potential, and there are these known benefits of diversity for groups and for fields, and also it's just fair. So I want to ask, are gender and sexual minorities actually underrepresented in STEM? And this is data that's really only become available recently, and I don't know what the situation is in Brazil, but in the US, there's now very good data for LGBT, lesbian, gay, bisexual, and trans employees in STEM-related US federal agencies. And these federal employees have legal protection from discrimination, so it's kind of a best-case scenario. And a giant survey of federal employees found that, yes, STEM agencies have fewer LGBT employees than expected, about 18 to 20% fewer than expected based on estimates for the general population, and about 13% fewer than non-STEM federal agencies. So why? They're legally protected. Could it be a hostile or chilly workplace environment, as has been argued for other minorities and for women? Well, LGBT employees in STEM agencies report worse experiences in terms of workplace fairness, in terms of perceived treatment, and in terms of job satisfaction, which is part of why we keep doing what we do. And this is compared both to their straight heterosexual colleagues in the same agencies, and also compared to LGBT employees in non-science agencies. And it's not mitigated by position in the organization, and it's not different by age or gender. So it's not just a bias for masculinity, and it's not just that it used to be bad and it's now getting better, so we can just wait. Yeah. Okay, what about a leaky pipeline? This is well documented for women in science. So in a broad national survey of students who began college, and again, this is in the US, as science freshmen, science, technology, engineering, and math freshmen, 4 years later, when they were approaching graduation, 8% of them identified as sexual minorities. Okay, so did this affect the likelihood that they're still in science? Yes. So there's a substantial drop of this people transferring out of science fields. The retention rate was about 6% lower for women than for men, and this is consistent with other studies that have been done. But the loss of sexual minorities was even greater. And there's also a sex bisexuality interaction effect. So sexual minority men were more likely to drop out of science than heterosexual men, and sexual minority women were more likely to persist. And this interaction modifies, but it does not reverse the effect of sex. And I say sex here deliberately because we have no comparable data on student gender identities. Okay, so how did I get from here to here? I'll tell you some parts of the story. So I did my undergrad degree in biology at the University of Guelph, and I figured out my queerness in second year. And I loved plant physiology. I took all the plant physiology courses there were, and I considered graduate school in that, but I was put off by a conversation with a professor in which he was really dismissive of the value of other fields, because I was interested in a bunch of things. Okay, I also took a bunch of women's studies courses, and I was a feminist activist, and I considered grad school in that, but I really didn't like indoor work, and really it wasn't much more queer-friendly than biology. So I could see no way to combine these two interests. So I just kept working in nature interpretation, which is what I had done as part-time and summer jobs through my undergrad. And I loved teaching people about nature, and I could be myself at work, but ultimately there was, at the time, no room to move up in that field, and I wanted more of a challenge. So one of the things that we did at the Nature Center was these awesome snake shows, where we would teach people about snakes. And I loved working with snakes. Their alien elegance and their misunderstood nature somehow appealed to my queer sensibilities. And I started thinking maybe I could study snakes. So I went back to school and I took the herpetology course that had not fit into my undergraduate schedule. And I learned about reproductive mode diversity in frogs, which basically blew my queer Canadian mind. I had no idea that range of variation was even possible in one vertebrate order. Because in Canada, you know, it's like this. The frogs go in the water and they drop their eggs there and then they go away. That's it. And in the tropics, frogs do all these amazing things, such a variety of ways to reproduce and live their lives. And looking at my career now, that hour of that lecture was probably the most influential hour of my life. Okay, and it gave me a path to continue in biology. Okay, so we all know that herpetology unites a queer combination of creatures, but it let me work on frogs and snakes, and the weirdness of the mix seemed to me potentially to make space for diversity of thought as a kind of interdisciplinarity internal to the field that had not been evident to me, for instance, in the mammalogy course that I had taken. So I went to Dalhousie University to do my master's degree with Richard Wassersug. And I studied tadpoles in a medical school, and I studied behavior in an anatomy lab. So my science was a little bit out of step with what people around me were doing. I was used to that from my personal life, no problem. And I published this work in 1992 in Copia. But before that, I contributed to this book, which is a groundbreaking anthology. It's been reprinted 3 times. It's on queer studies curricula. It's considered a catalyst for the bisexual rights movement and named one of the top 100 queer books of the 20th century. I used a pseudonym because I was applying to PhD programs in the US, and at the time, US immigration was denying entry to queer activists, so I did not want to take that chance. Okay, so I went to Texas. I went to Mike Ryan's lab to do neotropical field work, studying the behavior and integrative biology of frogs. But before I got there, I hadn't really thought about the fact that everyone else in the lab was studying sexual selection, right? How males compete for females and how females choose males to mate with. Okay. I did not want to make frog heterosexuality the focus of my dissertation. I was much more interested in natural selection, life and death, and I wanted to focus on early life stages, so the sex of my animals was both unknown and irrelevant. So in 1991, with a fellowship from Canada, I went to Costa Rica, to Corcovado National Park, looking for a frog with an interesting reproductive mode, interesting eggs or tadpoles, and a question for my PhD work. And I was introduced to the beautiful Agalichnus calidrius, which lays terrestrial eggs, super cool, on leaves over ponds, and then at hatching, the tadpoles fall into the water. And I started doing basic natural history, monitoring egg fates. All the little pink flags mark egg clutches, and what I saw was snake predation. A lot of snake predation. Snakes ate half of my monitored eggs. And I also had the experience of accidentally bumping into a well-developed clutch and seeing a few embryos hatch, which was actually pretty quick. So thinking about this, I came up with the hypothesis of adaptive plasticity and hatching timing. I thought maybe embryos can use hatching to escape from snakes, and predators in the water might select against early hatching if more developed tadpoles are less likely to be eaten. And then between field seasons, I presented this hypothesis along with my egg predation data and my planned experiments at a meeting. And some senior colleagues were very skeptical. They thought it was impossible for a frog to hatch fast enough to escape from a snake. And they thought it was impossible for a few days of development to affect the survival of tadpoles in the water. Well, of course, this was not the first time that people had advised me against pursuing my interests. So with Queer Resilience, I went back to Costa Rica. And I let snakes attack eggs in cages where I could watch. And I saw embryos hatch and escape. And I documented escape hatching at the pond, which was full of freshwater shrimp. And in my experiments, older hatchlings did survive better with these shrimp and with other predators compared to the younger hatchlings. So I was pretty psyched. I submitted this work for publication. And one reviewer wrote, "Every herpetologist knows that if you jiggle well-developed frog eggs, they hatch. It doesn't mean anything." They seem to think that the snakes were just inadvertently breaking already weakened egg capsules and releasing these passive embryos, right? And I had simply made a trivial misinterpretation. Okay, so just to be clear, I'm going to show you a short video. These embryos are 30% premature. The snake has already eaten 2 eggs. These 2 have ruptured their membranes, beginning the hatching process. And you'll see for this embryo, the whole hatching process in real time from decision to escape. Okay, so here's the snake. You can see it touching and moving the eggs as it feeds. This embryo is trying to escape. There it goes. Watch, this one's going to start wiggling. Okay, the egg's leaking. Trying to get out. A little bit of work. Okay, there it goes. Great. It escaped very unpassively. Okay, so we now know that these embryos store up hatching enzyme for rapid controlled release to make a small escape hole in the egg. And they do this in response to vibrational cues in snake attacks that they sense with the otoconal organs of their inner ears and also with their lateral line. And we're now working on mechanisms controlling when and also where they release the hatching enzyme. So this is a frontal view just after hatching, and the green marks where the hatching enzyme is. And we also now know that environmentally cued hatching is widespread in amphibians, and embryos respond to many different biotic and abiotic factors. And in fact, it's found throughout bilateria, and other people are now studying cued hatching directly and also using it to ask other kinds of questions. Like, how does hydration affect thermal physiology and the voluntary thermal maximum of embryos? This is not a question that I would have thought to ask, but Caroline Guevara Molina is asking it, and she's using hatching to do that. She's here at the conference and at Universidad de Sao Paulo. Her work is super cool, and you should ask her about it. Okay, so I think this kind of trajectory, from unasked question to impossible concept, to trivial misinterpretation, to isolated weird case, to motivating possibility, to general phenomena, to foundational knowledge for asking other questions. This is probably not unusual for novel ideas in science as evidence accumulates, right? But it highlights the importance of unasked questions and the fact that one person's impossible concept is another person's motivating possibility. And I doubt that I was the first herpetologist to accidentally bump into a red-eyed tree frog clutch and see an embryo hatch. And I know that we had known for a long time that some terrestrial amphibian eggs hatch when flooded. So why did I see a possibility where others had not? And what motivated me to pursue hatching as a potentially general phenomena? Well, I think that my migratory queer life has shaped my thinking and my interests. I know very well the experience of leaving a familiar but in some ways constraining environment for a different place with new possibilities and unknown risks, and of being balanced at that point of decision, weighing potential costs and benefits. And that experience has given me what my partner Andine calls imaginative microempathy for eggs and for hatching. And imaginative microempathy generates testable hypotheses. I'll give you one last example of something that I see as a motivating possibility. So these Hyperolias have individual spot patterns. And for his master's, Omar Grafe studied their reproduction. And he saw 7 of 24 females turn into males. So these were adult frogs that had already laid viable eggs, that had developed into tadpoles, and then they developed vocal sacs, and they started fighting with primary males, and then they amplexed females, and they fertilized eggs that went on to develop into viable tadpoles, and all 7 were in terraria with few or no primary males. And this was published 30 years ago. And some people have suggested, that it was a lab artifact, it was maybe endocrine disruption. And we know that chemical exposure during development can completely sex reverse frogs, so genetic males are phenotypic females, or vice versa. And it can also cause infertile intersexes, but these were fertile adult females. And females at even sex ratios with the same housing conditions, the same water, the same food, everything, did not change sex. So it seems to me that socially dependent sex change might be something that really happens, that this species does in nature. And maybe other frogs do this too. And of course, more work is needed to know the relevance of this in nature. But recent reviews have kind of dismissed this possibility, this evidence of functional hermaphrodism in frogs, because no further examinations were published in 30 years. But I would say that absence of evidence is not the same as evidence of absence. And if we consider the abundance of species and the diversity of forms and the number of evolutionary origins of hermaphrodism in fishes, as well as the ancestral reproductive similarities of these anamniote lineages, I think there should be some hermaphrodite frogs. And if there are not, there should be some reasons for that we don't yet know. And we know about the hermaphrodite fishes because people have done the work to find out. Okay, so Almar's frogs came from Tanzania. And I love East Africa and I have not been back in ages. But in the current legal environment, I will not be going there. Still, there might be other hermaphrodite frogs, and it would be super cool to find a lineage in the neotropics where we could study this. And in a broader sense, I would ask you to please do not underestimate the value of weird observations, either in the lab or in the field. Of course, it's not all you need, but weird observations can lead to testable hypotheses and bodies of work that transform our understanding of nature. And working with humans, different people can answer different questions because they have access to different human populations. And working with herps, anyone could potentially ask and answer any question. But also, our life experience does affect our interests and our perspectives, what we notice, what we imagine, what motivates us. And so we know that herpetology has made huge contributions to our understandings of sexual and life history diversity in nature. But also, without a doubt, undiscovered diversity remains. And we don't know how future discoveries may change our understandings, but we do know that sometimes what we think we know is wrong. And some unasked, unimagined questions will be transformative. So including diverse human perspectives in science, I think will facilitate new discoveries because different people ask and are motivated to answer different questions. And finally, it's well documented that women and minorities often face a higher burden of proof for their ideas. So do weird ideas. And you see the intersection. So for some of us, non-normative or queer lives helped prepare us to pursue science that at times runs counter to prevailing ideas. But also, support networks are crucial. And a lot of people have helped me over the years in many, many ways. And this is something that we all can do for each other. So that was both on a personal level and on a scientific level. So I want to thank all of my mentors, my advocates, my biological and chosen family for their support, as well as, of course, my funding agencies and all of my students, my collaborators and colleagues across disciplines and over the years. Thank you for listening.